everyone. In this class we will discuss about circular dichromism spectrophotometer. Okay, so let's begin with the class. So what is circular dichromism spectrophotometer? So uh, Circular dichromism spectrophotometer is a form of light absorption spectroscopy. Like other spectrophotometer, it is also a light absorption spectroscopy. Okay, and that measure the differences in absorbance of right as and left circularly polarized light. Okay, rather than the commonly used absorbance of isotropic light by a substance which arises due to its structural asymmetry. Okay, so what is happened is in the circular dichromism spectrophotometer, it absorb the circularly means right handed, okay, or you can say like clockwise direction moving light as well as the anti-clockwise moving light. Okay, so this is the right handed circularly polarized light, and this is the left handed circularly polarized light. Okay, so what when you passes a light, then according to the structural asymmetry or the chirality property of any kind of molecule, what it will do is, according to its preferences, it will rotate the light either in circle means right handed direction or in the left handed direction. Okay, and the percentage between means one light it will rotate in right handed direction, and the percentage rotated in the left hand direction, that difference it is measured by the circular dichromism spectrophotometer, and based on this property it can identify either the tertiary structure as well as the secondary structure of the protein. Okay, so it is depend on the chirality, or it is depend on the substance is asymmetrical appearance. Okay, so nature. So the absence of regular structure result in the zero CD intensity, while an ordered structure result in the spectrum which can contain both positive and negative signals. Okay, so if there is no regular structure, suppose there is a symmetry, okay, there is no chirality, then what will happen is the CD intensity what you will get is zero. Okay, so if you are getting the CD intensity zero, it through that you can identify like your molecule is denatured. Okay, it is not properly folded, it is not having proper secondary structure or proper tertiary structure. Okay, so then only you are getting the zero CD intensity, while an ordered structure, if it is ordered structure, if it is properly folded, then you will get positive as well as the negative signal. Okay, then it has been shown that CD spectra between 260 and approximately 180 nanometer can be analyzed for a different secondary structural type. Either it is alpha helix, palar, and antipalar beta sin, turn, and others. Okay, so this is the optimum range, like 260 to 180 nanometer. You can find out the different secondary structural component like alpha helix, palar, antipalar beta sheet, and turn. Okay, when you ever go down below 180 or 178, that time you can identify the more different kind of structures, but means most of the time we are unable to go beyond because of the whatever buffer we are using, whatever protein we are using, that create noise after certain nanometer difference, it will create noise. Okay, so that's why we are unable to means take the those structural property, but this is the standard range, and in that standard range you can identify the alpha helix, palar, antipalar beta sheet, turn, and other secondary structure also. Okay, then how many different type of secondary structure can accurately be distinguished is highly dependent on the wavelength range of the spectrum. Okay, as I told you, like as you go beyond, you can find out that much different kind of secondary structure. Okay, and if you are here only, then you can only identify like alpha helix, beta sheet, and turn. Okay, but if you go beyond, you can find out the more kind of structural secondary structures. Okay, so in the data between 200 to 260 nanometer, the information content is 2. Okay, so if you are taking the spectra between 200 to 260 nanometer, then it will give you only two kind of secondary structure. Okay, information alpha helix and beta sheet. Rising to 3 if data down to 190 nanometer are used. Okay, then if the data extended down to 178 nanometer, there will be 5 secondary structure. Okay, and if you go more beyond, then it will give you the about 8 secondary structural content. Okay, so that you have to remember, like 200 to 260 nanometer, it is giving two type of secondary structure. If you go beyond 190 nanometer, then it will give three secondary structure including random coil. Okay, and when you go beyond extend down to 178 nanometer, the value is 5. Okay, and by taking a spectra of 160 nanometer, it will give you the 8 different secondary structural content. Okay, then depending on the light source used for the generation of circularly polarized light, there are three different type of CD spectroscopy. One is the far CD UV-CD spectroscopy. Okay, and second one is the near UV-CD spectroscopy, and third one is the visible UV-CD spectroscopy. Okay, so the far UV-CD spectroscopy is used for the secondary structure property identification. Okay, and the near UV CD spectroscopy is used for the tertiary structure identification, and this visible CD spectroscopy is used for the metal ion protein interaction. Okay, so yeah, basically we use this far UV CD spectroscopy, okay, for the secondary structural structural study of the protein. Then near UV CD spectroscopy is used for the tertiary structure protein. Okay, so these are most commonly used CD spectroscopy that we not use that much frequent, but through visible CD spectroscopy you can identify like metal and protein interaction. Okay, so this classification is based on the light source used for the generation of circularly polarized light. Okay, so how it works, like the principle is, when you monochromatic light from CD1 is passes through a photoelastic modulator. Okay, this is the monochromatic light. When you passes through this photoelastic modulator, what it will do is which convert the linear polarized light into alternating left and the right handed polarized light. Okay, so the two polarization are differentially observed, and the difference in the observation is detected with a photo multiple tube. Okay, so what is happening is, suppose this monochromatic light when passes through this photoelastic modulator, it gives 50% circular means left handed polarized light, and 50% right handed polarized light. Okay, so according to this substance feature or the substance characteristic, what it will do is, either it will observe more right handed polarized light or the left handed polarized light. Okay, so then whatever transmission happen here, the difference means certain left-handed polarized light will difference transmit, and certain right-handed polarized light will transmit. Okay, so that difference is calculated by the CD signal. Okay, the difference of left-handed polarized light and right-handed polarized light. Okay, because here you are giving 50% of each type of light, and according to the this substance preference, it will observe either right-handed polarized light or left-handed polarized light, and then transmission happen accordingly, and that whatever difference it will comes here, it is identified by the CD signal. Okay, suppose here it is observed mostly 40% out of 50%, it is observed 40% of light, only 10% light is transmit, okay, of right-handed this polarized light. Okay, whereas this left-handed polarized light is 40% transmit. Okay, only 10% is absorb. Okay, so this signal will give you like 40 minus 10. Okay, so this will be 30. Okay, that signal is identified as a CD signal here. Okay, so there are in the graph, in the detector there is two type of signal is observed. One is the absorbance, like on a particular wavelength it will giving you a one kind of signal. Based on that you can find out the secondary structural content. Okay, then this is the HV, this is nothing but voltage suit up. Okay, so like if you are like your in your sample there is a certain kind of molecule also present which can cause the noise. Okay, so that is represented by this voltage suit up. Okay, so there is two type of graph, one is the CD signal and another one is the HV. That HV is for the noise signal and this is for the CD signal. Okay, then here you can find out different type of CD spectra. Okay, so this you have to remember, like if your sample is having the aDNA. Okay, if your sample is having the aDNA, it means the characteristic feature of the spectra of aDNA is 260 nanometer positive peak you can find out. Okay, so this is the like aDNA. Okay, here. Okay, this one. So at 260 nanometer there is a positive peak, and then 210 nanometer there is a negative peak. Okay, here 210 nanometer there is a negative peak, and then again 190 nanometer there is a positive peak. Okay, so there is two positive peaks at a 190 nanometer. Okay, and another one is the 260 nanometer, and one negative peak as a 210 nanometer. Okay, so that you have to remember, that is the characteristic spectra of aDNA. Then bDNA there is 275 nanometer positive peak. Okay, so 275 nanometer there is a positive peak. Okay, so yeah, just a second here. Okay, so 275 nanometer there is a positive peak, 240 nanometer there is a negative peak. Okay, 258 nanometer there is a crossover. Okay, this is the crossover. Okay, so this is the bDNA characteristic feature. Okay, there is another type of DNA which is called as the Z form of the DNA. Okay, and this Z form of the DNA is having a negative band at 290 nanometer. Okay, here it is the 290 nanometer negative peak. Okay, and negative band at a 290 nanometer. Then you are having sorry, here it is the 290 nanometer negative peak is here. Okay, and positive peak is present at a 260 nanometer. Okay, so this is the positive peak at 260 nanometer, and crossover is present as 185 nanometer. Okay, so this is the cross over about 180 nanometer. Okay, Z form is not the minor image of B form. The blue shift of the 200 nanometer of B form of the 185 nanometer is the Z form appear to be the trademark of B to Z transition. Okay, so if you will see here, as when you compare the bDNA to zDNA, here you can see that the crossover as well as the negative peak, here it is the negative peak is 240 nanometer, whereas here the negative peak is 290 nanometer. Okay, and the positive peak here you can see here, 200 nanometer, and here 185 nanometer. Okay, so it is causing the blue shift. Okay, here it is causing the blue shift. Blue shift means it is moving your left side. When it is moving toward the right side, then it is the red shift. Okay, so red shift means your protein molecule is more open towards the polar solvent, and if it is blue shift, it is your molecule is buried in nature. Okay, very compact, it is not very much interacted with the water molecule. Okay, so yeah, this is about the zDNA. Then you are having the proteins secondary structural content information. So first one is the alpha helix. So in the alpha helix, if you have seen, like negative band at 222 nanometer and 208 nanometer, okay, and positive band is 193 nanometer, okay. So here if you will see, there is a two negative peak, okay, one is 222 nanometer and one is 208 nanometer, okay, and one positive peak as 193 nanometer. Then beta helix have negative peak at 218 nanometer and positive band at 195 nanometer, okay. So this is the this red color is the beta sheet. There is one negative, one positive peak, okay. At 218 there is negative peak, and to 195 there is a positive peak, okay. Then you are having the random coil. Protein lacking any ordered secondary structure will not have any peak above the 210 nanometer, okay. So here it will not having any peaks apart from the 210 nanometer. There will be no peak, okay, it will be flat, okay. So this is the 210 nanometer random coil, okay. So this is three different type of secondary structural content spectra, okay. So alpha helix is having 208 and 222 nanometer negative peak and 193 positive peak. Beta sheet is having 218 positive peak and 218 negative peak and 195 positive peak, okay. Then random coil is 210 nanometer apart, there is no peak, okay. Then you can say like your protein is having no second means ordered structure, okay. So yeah, so based on that background, you are having one question like the circular dichromism spectra of the three protein PQR are given below, okay. So this is the three different type of protein, choose the correct combination, okay. So here if we will see this graph, here it is having the two negative peaks, okay, and two negative peaks. Here first of all it is saying about the proteins, okay. So don't recall the spectra of nucleotide, nucleic acid, that is the DNA, A form, B form and Z form of the DNA, okay. So here you have to just focus on the protein spectra, okay. So there are three different type of protein spectra we have seen: alpha helix, beta sheet and random coil, okay. So this is looks like the alpha helical spectra because it is having two negative peak at 222 and 208, as well as there is a positive, little bit positive peak, that is the 193 positive peak, okay. So that is the characteristic feature of alpha helix, okay. Here you can see that there is two negative bands and one positive peak is here, okay, 193 nanometer. So this is the alpha helix, okay. Then alpha helix, there is two option, P is having alpha helix, okay. So this option B and option D will be cancel out, okay. Only these two will be your correct answer, okay. Out of these two only one will be your correct answer. So P will be alpha helix. Then we will move to the Q, okay. So Q is having, if you will see here, Q is having one negative peak at 2 something 218 nanometer, okay, and here 195 nanometer there is a positive peak. So this is the characteristic feature of beta sheet, okay. So here like this, okay, this one, this red color, okay, have negative band at 218 nanometer and positive band at 195 nanometer, okay. So this will be the beta sheet, not the alpha uh, not the random coil, okay, because random coil we have seen that after 210 nanometer there will be no peak, okay. So this is the after 210 nanometer there is no peak, okay. Then you can say like it is the random coil, okay. So this is the third R will be your random coil. So the correct option of this question will be option A, that is the P will be your alpha helix because it is having two negative peak and one positive peak, Okay, and beta seat is having one negative, one positive, okay, at 218 and 195. Then random coil after 210 there is no peak, so it is the random coil. Okay, so this is the option A will be your correct answer. So thank you.